Cloning, the process of creating genetically identical copies of organisms, has captivated scientific and public interest for decades. Today, we will delve into the intricacies of cloning, including its history, methods, applications, ethical considerations, and potential future developments. To grasp the significance of cloning, let's explore its historical context and key milestones. The concept of cloning traces back to ancient times when humans noticed the ability of plants and simple organisms to reproduce asexually, generating identical offspring. Significant advancements occurred in the 20th century, particularly with the elucidation of DNA structure and the discovery of genetic principles. In 1953, James Watson and Francis Crick unveiled the double helix structure of DNA, providing a foundation for understanding inheritance and genetic manipulation. In 1996, researchers at the Roslin Institute in Scotland successfully cloned a sheep named Dolly using somatic cell nuclear transfer, SCNT, revolutionizing the field of cloning. Dolly's birth marked the first instance of cloning a mammal from an adult somatic cell, highlighting the potential for genetic replication. Cloning encompasses various techniques, each with its unique approach. Let's explore the primary methods employed in cloning. Somatic cell nuclear transfer, SCNT. SCNT, the most well-known cloning method, involves the following steps. Donor cell selection. A suitable donor organism is chosen from which the genetic material will be derived. This organism may be a multicellular plant, animal, or human. Egg cell collection. An egg cell or oocyte is obtained from a female organism of the same species. In some cases, the egg cell's nucleus is removed, creating an enucleated egg. Nucleus extraction. A somatic cell, typically sourced from the donor organism, is obtained. This somatic cell harbors the complete genetic material, DNA, of the organism. Nuclear transfer. The nucleus of the somatic cell is extracted and introduced into the enucleated egg cell. This can be achieved through nuclear injection or cell fusion techniques, such as electrical stimulation. Activation and development. The reconstructed egg cell, now containing the donor's genetic material, is stimulated to initiate cell division and development using chemical triggers or electrical cues. Implantation or in vitro development. In animal cloning, the resulting embryo can be transferred to the uterus of a surrogate mother for implantation and further growth. In the case of plants, the embryo may be cultivated in a suitable environment. However, it is crucial to note that human reproductive cloning is ethically and legally prohibited in most countries. Apart from SCNT, alternative methods like embryo splitting, artificial embryo twinning, and gene cloning, recombinant DNA technology have been utilized in specific contexts and research settings. Cloning has far-reaching implications across numerous fields, with potential applications and benefits, including reproductive cloning aims to create genetically identical individuals. While successful in animals, the cloning of humans for reproductive purposes raises significant ethical concerns and is widely considered unethical and prohibited in most jurisdictions. Also known as research or therapeutic cloning, this approach involves generating embryonic stem cells for medical purposes. These cells can be used to study diseases, develop treatments, and potentially regenerate damaged tissues. Therapeutic cloning holds promise for personalized medicine and regenerative therapies. Cloning can contribute to conservation efforts by creating genetically identical individuals of endangered species. Additionally, it has the potential to enhance agricultural practices by producing high-yielding or disease-resistant livestock and crops. Cloning provides researchers with valuable tools for studying genetic diseases, cellular development, and the role of specific genes. Animal models created through cloning have played a pivotal role in advancing scientific knowledge in various fields. The ethical dimensions of cloning have sparked profound debates and contemplation. Several key considerations include: the cloning of humans raises fundamental questions about human dignity, individuality, and the uniqueness of human beings. Cloning for reproductive purposes is often criticized for disregarding the inherent value and distinctiveness of individuals. Cloned organisms may face health risks such as developmental abnormalities, compromised immune function, or premature aging. Ensuring the well-being and quality of life for cloned individuals remains a critical concern. Cloning could potentially have significant psychological and social consequences for cloned individuals, their families, and society at large. Issues of identity, family relationships, and societal acceptance are central to these considerations. Cloning challenges moral and religious beliefs regarding the creation of life and the boundaries of human intervention in nature. Different cultural, ethical, and religious perspectives influence public opinions and acceptance of cloning practices. Cloning continues to evolve, and ongoing research holds promise for advancements in the field. Potential areas of future exploration include: efforts are underway to enhance the success rate and efficiency of cloning techniques, reducing health risks and increasing viability. Continued research in therapeutic cloning may lead to breakthroughs in regenerative medicine, with the potential to revolutionize treatments for various diseases and injuries. As cloning technology progresses, the development of robust ethical guidelines and regulatory frameworks becomes increasingly important to ensure responsible and accountable practices. Cloning represents a remarkable achievement in genetic engineering, with profound implications for various domains of science and society. By understanding the complexities of cloning, its methods, ethical considerations, and potential future developments, we can engage in informed discussions and shape responsible policies that balance scientific progress with societal values and ethical principles. Cloning is a subject that continues to captivate scientists, philosophers, and the public alike. As research progresses and societal perspectives evolve, the profound implications of cloning warrant ongoing exploration, dialogue, and critical evaluation.